Hello, everyone. When I was presented with the task of finding a peer-reviewed article that connected to my research project, I knew that I wanted to incorporate the liver somehow into the perimeters of my presentation, so I aimed to find an article that focused on a way to improve liver function from damage due to alcohol. And after some research, I found an article that uses zebrafish as a model organism to inhibit vascular endothelial growth factor, known as VEGF, to facilitate liver repair from acute ethanol-induced injury. Okay, so I'll start by telling you a little bit about this article. So the title of the article is Inhibition of Vascular Endothelial Growth Factor Signaling Facilitates Liver Repair from Acute Ethanol-Induced Injury in Zebrafish. Okay, so this article uses zebrafish as a model organism to help determine the role of vascular endothelial growth factor and its receptors by blocking their pathway. So why would we want to do this? What would this tell us? What would this prove to us? So it's thought that blocking the vascular endothelial growth factor can be used as a therapeutic measure to enhance liver repair. So actually, it was found that chronic liver disease upregulates VEGF, which in turn leads to angiogenesis, and which is the formation of new blood cells from pre-existing blood cells. So, one key term in this study is going to be hepatic stellate cells, or HSCs. These cells generate scar tissue and upregulate the vascular endothelial growth factor in their receptors. So the study uses translucent larvae from the zebrafish because they're easy to view, and it allows us to be able to see the effects of alcohol better. So this study exposed 4-day-old post-fertilization larvae to 2% ethanol for 24 hours. And why they chose that time frame, that age, would be because 4-day-old post-fertilization larvae, that's the minimal age where the liver is optimally functional. And it has, at that age, they have the enzymes necessary to metabolize ethanol. So this induced hepatic stenosis and HSC activation. So why is this research important? So alcoholic liver disease, or ALD, is characterized by chronic or acute alcohol consumption. So alcoholic liver disease progresses systematically. So it progresses in chronological order. It starts with hepatic stenosis, which is fatty liver disease, where too much fat accumulates in the liver, in the hepatocytes of the liver. And then from there, individuals may develop alcoholic hepatitis, which is inflammation of the liver. And then lastly, liver cirrhosis is the irreversible scarring of the liver that leads to long-term damage and impaired liver function. So this research is useful because it examines a way to make repairs to the liver after damage from alcohol exposure or consumption. Okay, so now that we're familiar with the direction that this article is going in, let's talk about the hypotheses. So the hypothesis is, when inducing hepatic stenosis in zebrafish, inhibiting the vascular endothelial growth factor can play a role in regulating sustained liver injuries after acute alcohol exposure by mediating interactions between activated hepatic stellate cells and blocking KDRL to make developments in alcoholic liver disease therapeutic remedies. So I know that was a lot to digest, so let's simplify it a little bit. So this study exposes zebrafish to alcohol, which induces hepatic stenosis. From there, they suspect that inhibiting the vascular endothelial growth factor will enhance or repair injury to the liver. So to evaluate this, the researchers examined hepatic stellate cells and predicted that their exposure to ethanol would increase the expression of VEGF. It was thought that KDRL would be expressed when exposed to ethanol, therefore inhibiting KDRL can suppress angiogenesis and HSC activation. So angiogenesis goes hand in hand with fibrosis and cirrhosis of the liver. Those promote the progression of alcoholic liver disease. So if we determine the roles of VEGF and HSC and KDRL jointly, this can allow us to develop therapeutic solutions to stop the progression of alcoholic liver disease. Okay, here we have figure 5 to the left, and we have A, B, C, and D, where the green and black images are. And this shows a 3D projection of the liver's intrahepatic vasculature for a wild type and a KDRL 19 mutant at 24 hours post-fertilization. And the KDRL mutant and wild type have similar number of HSCs in the control group, so A and C. And then if you look at B and D, there is decreased fibrosis observed in the ethanol treated with the KDRL mutant seen in D, and the ethanol treated wild type shown in 5B, then the wild type B. And this is indicative of impaired angiogenesis, which is visible by fewer hepatic branches. And then if you look to the right, in 5E, the bar graph, they evaluated the thought that the ZM treatment acts on multiple VEGFRs, those receptors. So to confirm this, an ethanol treatment was used on the KDRL 19 mutant that carries an exon deletion on its gene, and this mutant exhibited milder phenotypes in terms of intrahepatic branching, if that were to be true, in which you can see the blue is the KDRL mutant, and those lines have less branching, so it was proved to be true. And the wild type and KDRL gene exhibited similar branching, which was expected and predicted. So the branching correlates to angiogenesis, and the mutant kind of inhibited the excessive branching. Okay, here we have figure 7B, which here a 2 micron ZM treatment, which is used to inhibit KDRL, block VEGF, and suppresses hepatic steatosis, and increases the HSC number at 27 HPT. So this leads to the conclusion that steatosis and HSC proliferation can be suppressed by VEGF inhibition. And the important part to note here is that if we can successfully find a way to inhibit VEGF, then we have a way to stop angiogenesis. So why is that important? Well, in cirrhosis and fibrosis of the liver, new blood vessels are constantly forming, which is the definition of angiogenesis. And they're forming at an exponential rate, and it's depleting our all of our other healthy organs from the oxygen and nutrients they need because the blood flow is redirected towards the liver, and there's an increased blood supply there at the site of the disease. So angiogenesis actively contributes to the metastasis of alcoholic liver disease when VEGF is upregulated. So here we found a way to inhibit VEGF, which is a way to stop the progression of alcoholic liver disease. Okay, so here are the most important conclusions from the findings. So VEGF activates HSCs after exposure to ethanol, which leads to steatosis in hepatocytes. Due to the fact that KDRL mutants didn't develop hepatic stenosis, steatosis at 0 or 27 HPT, it can be said that KDRL could be an obligatory effector for ethanol-induced hepatic steatosis. And in other words, basically what this means is it's required to induce the disease state. That's what a obligatory effector is. So HSCs were found to maintain the extracellular protein matrix deposition and expression of fibrogenesis genes to promote HSC proliferation. So this explains why the KDRL mutant showed signs of fibrogenesis after the ethanol exposure. And on the flip side, hepatic steatosis can only be stopped when KDRL is inhibited. It was also found that HSCs and endothelial cells expressed KDRL after acute ethanol treatment, as expected. So through my research and analysis, I also concluded that the ZM treatment could be a beneficial treatment option to explore, and ideally an anti-VEGF agent would be a potential candidate for a remedy to alcoholic liver disease. So these findings can definitely be further analyzed to more clearly understand how we can further explore alcoholic liver disease and hopefully find some therapies in the future. Okay, so what do the future implications look like for this study? So this article gave me some insight into the way that zebrafish metabolize alcohol, and it also increased my knowledge on the pathways involved in these processes. And zebrafish are good for imaging because their bodies are transparent almost, and their larvae as well. So it's fairly easy to observe other body systems in the fish if additional research were to be conducted on these model organisms. And we're also able to do live imaging, like just in our class already. We're taking videos of our fish and observing different, tactile responses and things as such. So we know that it's easy to do live imaging and videoing on these organisms. And the article did make mention that they plan to further their research by determining the origin of the myofibroblasts and investigating more into the VEGF signaling pathway. And zebrafish are great for studying alcoholic liver disease and conducting research on that, because in humans it takes years and years of alcohol consumption to basically destroy our livers and get it to the point where it's not optimally functional. And in zebrafish we can get those same results in about a day. So it's quick and easy, it's effective, and it's also easy to collect hundreds, even thousands of embryo weekly. So this gives hope to novel alcoholic liver disease therapy discoveries in the future, because we're able to do, mass trials, and we're able to crank out a bunch of numbers quickly and easily. And one direction that needs a little bit of focus is developing a screening process and tools for identifying genetic mutations within the fish. And it's also unclear, the study mentioned that it's unclear if the fish are experiencing chronic or acute liver disease. So there is room for improvement for the further to in future improvement and further development with this study, but there are high hopes for some groundbreaking discoveries, and hopefully we'll know more about alcoholic liver disease in the upcoming years. Okay, so how can this information be applied to my personal research project? So in my own research project, I expose my zebrafish at 24 to 36 hours of age, but in the article they expose at 4 days of age, and this is because I found out that through the article that the livers of the fish aren't fully developed and they don't have the enzymes to metabolize alcohol until 4 days post fertilization. So I could use the same liver repair therapies in my own research to determine if an earlier or later time point would be more effective. So if I expose at 24 hours like I do in my experiment, if that would change the liver repair mechanisms, how effective they are, versus 4 days when the liver is fully developed. And I hypothesized that if I were to do this, I would hypothesize that the liver repair mechanisms would be less effective on the less developed or the younger fish. So I think it would be interesting to observe that. And also, take it a step further, I was thinking that it would also, I'd be curious to know the effects of the offspring of the fish that were exposed to ethanol. So, say a female is exposed to ethanol, allow her to mate, lay eggs, and then check the her embryo for signs of like fetal alcohol syndrome, or specifically like liver abnormalities, and then from there, if they do have similar abnormalities as the mother did, then testing the same liver repair mechanisms used on the adult fish. And I think that would allow us to make a connection between ways to reverse or improve the symptoms of those affected by fetal alcohol syndrome. And I believe that all new knowledge is useful in increasing our understanding of the zebrafish, and extrapolating that out to humans through our similar gene pool. So I think that would be interesting to evaluate in my study. And lastly, this is the article reference, and thank you for watching.